Hello everyone. Let's continue to study Section 2, Cellular Adaptations. Last time we talked about adapted cell hypertrophy and hyperplasia. Now let's move to atrophy and metaplasia. The definition of atrophy is shrinkage in the size of the cell by the loss of cell substance, usually accompanied by decrease in the number of cells. That happens in normally developed organ or tissue. Here, we need to distinguish it from hypoplasia and aplasia. Hypoplasia refers to organ or tissue that cannot develop fully to normal size. Aplasia refers to organ or tissue in a state of non-developing. Hypoplasia and aplasia are all congenital defects. Atrophy can also be divided into two types: the physiologic atrophy and pathologic atrophy. The pathologic atrophy, here we take brain as an example. Picture A is the brain of a 82-year-old male. Picture B is a normal brain of a 36-year-old male. We notice that the physiologic atrophy occurred during aging process. The size of brain decreased and the gyrus became narrow. Sulcus became deeper and wider. The pathologic atrophy, there are 5 common causes. Let's go through them one by one. The first one, inadequate nutrition. This is a patient with malignant tumor, the liver cancer. Because cancer cells compete for nutrition with human body, that results in cachexia. In this picture, we notice that the patient became very thin, but with a big belly, that is ascites. Lots of fluid in peritoneal cavity, that is generalized inadequate nutrition. As for local inadequate nutrition, we take the cerebral atherosclerosis as example. Once the cerebral vessel is narrowed by atherosclerosis plaque, just like this one, and this one. The blood supply will decrease to cause inadequate nutrition in local brain, which gradually undergo atrophy. The second cause of atrophy, decreased workload. Look at this picture. The man is lying in bed. Because of fracture, the patient is restricted to complete bed rest for at least 1 to 2 months. The workload of skeletal muscle decreased. Then comes the atrophy of muscle. The third cause is loss of innervation. Because of poliovirus infection, the neurons in the anterior horn of the spinal cord were injured. The innervation state of lower limb result in muscular atrophy. As we see in this picture, the leg become thinner. The fourth cause of atrophy, pressure. In this picture, a brain with metastatic melanoma nodules. There are at least 3 nodules on the cut surface. The red arrows point to enlarged tumor nodules, which cause atrophy of the surrounding brain tissue. This is a case of hydronephrosis. Obstruction at the renal pelvis or ureter may cause urinary retention. The persistent pressure of retained urine in the pelvis will cause kidney atrophy. In this picture, the renal pelvis is obviously enlarged. The cortex and medulla become thinner. The fifth cause of atrophy, loss of endocrine stimulation. Many endocrine glands, the breast and the reproductive organ, are dependent on endocrine stimulation. For example, the breast and reproductive organ undergo atrophy in postmenopausal women. The histological changes of atrophy. These atrophic cells have lower level of protein synthesis, so the size and number of cells both decreased. In many situations, the number of autophagic vacuoles will increase. These vacuoles are undigested organelle residual in autophagy lysozyme. We call them lipofuscin. They usually appear in the atrophic heart or liver. Lipofuscin granule is brown, located in the perinuclear area, just like here and here. The upper picture is normal myocardium, and the lower picture is atrophy of myocardium. Can you tell the differences between them? Well, the atrophic cells become smaller and wavily arranged, with light brown color. This is atrophic skeletal muscle. The number and size of cell decreased, like here and here. So the mesenchyma is broadened. The fat and connective tissue filled in the space around atrophic skeletal muscles. The fat tissue, fat tissue, fat tissue. So the total volume of tissue is not significantly reduced, but the muscle strength is weakened. We call it pseudohypertrophy. Now let's move to the last form of adaptation, metaplasia. Its definition is a reversible change in which one adult cell type, the epithelium or mesenchyma, is replaced by another adult cell type. Here, we must know that metaplasia is thought to arise by genetic reprogramming of stem cells, but not transdifferentiation of those differentiated cells. This is a picture of columnar epithelium and basal membrane. This is columnar epithelium. These cells are basal cell, also named reserve cell or stem cell. We know that the stem cells have the ability to differentiate into particular cell lineage. They act as supplement. When the columnar epithelium is damaged or lost, they will differentiate it into new columnar epithelium. But on the right side of this picture, the epithelium changed into squamous cells. How did that happen? Well, the columnar epithelium cannot directly change into squamous epithelium. Normally, because of the persistent pathologic stimuli, the former columnar epithelium were destroyed. Then, the basal cells were reprogrammed to differentiate into squamous epithelium in response to the external stress. Metaplasia is caused by chronic stimulation. Chronic stimulation can activate or suppress some related genes. They function via signals generated by cytokines, growth factors, and extracellular matrix components in the cell's environment. Squamous metaplasia is one type of epithelial metaplasia and is usually seen in respiratory tract. It is in response to chronic irritation, deficiency of vitamin A, cigarette smoking. It can be observed in the respiratory tract of smoker. This is a normal ciliated columnar epithelium located in trachea. Here, stratified squamous epithelium replaced the ciliated columnar epithelium. Because of chronic injury or chronic irritation, we call this squamous metaplasia. It is a reversible change. Once the chronic irritation abates, they return. Here comes the question. Is the squamous metaplasia in trachea good or bad for our body? Well, it's a double-edged sword. In one hand, the squamous epithelium has more survival advantage than other type of epithelium because of its multiple layers of cells. They can tolerate even more harsh conditions than columnar epithelium. But, on the other hand, the function of ciliated columnar epithelium is lost. We know the ciliated columnar cells can secrete mucus and help to clear the respiratory tract by ciliary swing. Sometimes, if the chronic irritation persists, malignant transformation could be induced. Squamous epithelium in the lower esophagus may transform into gastric or intestinal type columnar epithelium. It usually occurs in gastric reflux. In this picture, here is the squamous epithelium of esophagus. But we can see from this side to the left, it's replaced by columnar cells. We call this columnar metaplasia. Metaplasia may also occur in mesenchymal tissue. Bone or cartilage is occasionally formed in soft tissues, particularly in foci of injury. This is called bone metaplasia or cartilage metaplasia. For example, in this picture of injured muscle, the red circles are skeletal muscles. Some of them are replaced by cartilage. Here, in the white circle, that's cartilage metaplasia. But one thing is quite different here. Unlike the reversible metaplasia in epithelial tissue, once the bone metaplasia and cartilage metaplasia are formed. They cannot be restored, even if the chronic irritation is gone. Well, it's the end of cellular adaptations. Let's take a summary. Number 1, we learned what adaptation is and the 4 types of adaptation. They are hypertrophy, hypoplasia, atrophy, and metaplasia. Here are some questions for you. Thanks for watching.